Namaste students, I am JV Dr. Aziz Omithan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth Women's University, Jaipur. Today I am going to discuss about the topic history of the microbiology, which is the topic which belongs to the uh, fundamental of the microbiology. What is the history? In previous video, I discussed about the microorganism and the microbiology. What is the microbiology and what are the microorganisms? And on microbiology, the branch which deals with the microorganisms. So the first time in the microbiology, the scientist who developed some theories is the Anthony von Leeuwenhoek. Anthony von Leeuwenhoek was the first to observe living microorganisms. He was the first scientist to observe the living microorganism. He he developed a single lens magnified 50 to 300x magnification lens. Okay, lens with magnification of the 50 to 300x magnification value. And between the 1674 and 1723, he also wrote a series of the papers which describe about the observations of the bacteria, algae, protozoa, and fungi, which are the which he called it the animals. And Robert Hooke was the second one. He was the one of the most important um, discoverer in the field of the microbiology. And in 1665, the crude microscope, Robert Hooke stated that the life's smallest structure unit is the cell, and it was the greatest discovery in the field of the biology, in which Robert Hooke declared that the smallest structural unit is the cell, and it is the basic unit of the life. Then the second discoverer was Louis Pasteur between the 1822 to 1895. And Louis Pasteur developed the germ theory in 17. He developed the germ theory. He also developed the vaccines and the anthrax. He used a technique which is known as the pasteurization technique, and he developed the germ theory of the disease. He is known as the father of the bacteriology and the immunology. He, he is known as the father of immunology and the modern immunology. In 1864, Pasteur developed a relationship between the microorganism and the disease in preventing wine from the spoilage and using the process which is known as the pasteurization. And this process kills bacteria and the alcohol by heat, and it is used for the prevention of the formation of the acetic acid. And by using this pasteurization technique, formation of the acetic acid will be reduced, and when it will be reduced, then the food can be preserved. He discovered the pasteurization lead to the introduction of the germ theory of disease in 1864, and he stated that the disease are caused by the growth of microorganisms in the body. Okay, but people were thinking that the disease are caused by the sins. Disease are caused by the bad characters. Disease are caused by the poverty, not by the microorganisms. So this pasture proved them wrong, that that the disease are caused by the microorganism. It is not by your good and bad characters. It is not by your sins. It is not by your poverty. It is by it is by your, it is by the microorganism. Next sentence is the Robert Koch. Robert Koch developed an elaborate technique to identify the pathogens that cause specific disease. He isolated a bacteria which is Bacillus anthracis, cause anthrax disease, and he developed the germ theory of the disease. And in 1876, Robert Koch proved that the germ theory of disease by showing the bacteria actually cause disease. Koch established a sequence of experiment that directly relate to the specific microorganism to a specific disease called Koch postulate. He developed some pure culture. He identified some bacteria such as the Bacillus anthracis who caused the anthrax, Mycobacterium tuberculosis who caused TB, and Vibrio cholerae. Alexander family Fleming was the next scientist, and in 1928, Fleming observed that the microorganism, the growth of the bacterium Staphylococcus aureus was inhibited by the some areas. It was inhibited in the areas surrounding the colony of the mole that had contaminated a petri dish. That the mole was identified as a penicillin notatum. So he came to know that every microorganism protect itself against some another microorganism. So by this theory that the Staphylococcus aureus was inhibited by the penicillin notatum, then he developed a vaccine which was known as the penicillin. Example of some history background in food microbiology also. In 700,000 BC, evident that the Babylonian manufactured beer, and one appeared in about 3500 BC in early civilization. Uh, in underdeveloped countries where modern sanitation is lacking, alcohol beverages like beer and wine were much safer to consume than the local water supply because the water was often contaminated with intestinal microbes that cause cholera, dysentery, and other serious diseases. In 3000 BC, Egyptians manufactured cheese and butter. Okay, they manufactured cheese and butter by the fermentation method. Okay, then again fermented foods such as the cheese and sour milk, yogurt was also developed, and it was safer to eat and resisting spoilage better than the raw agriculture counterparts. Several cultures also learned to use salt to preserve meat and food. So they were using salt so that the food can be preserved. And in 1000 BC, the Romans used snow to preserve shrimp because the low temperature microorganisms cannot grow very well. And they also use smoked meat. In 1665, in Italian physician Francesco Redi, he demonstrated that the mangoes on fortified meat did not arise spontaneously, but were still the larval stage of the flies. This was the first step away from the doctrine of the spontaneous generation. In 1765, an Italian named the Spallanzani, he tried to disprove the theory of the spontaneous generation of life by beef, demonstrating beef, okay, demonstrating beef broth, which was boiled and then sealed, remained sterile. His work was criticized because he believed oxygen was excluded. Okay, and which they thought was vital to spontaneous generation. And in the 1795, the French government offered 12,000 francs to anyone who could develop a practical way to preserve food. Then a French confectioner who was named Nicolas Appert, he issued the patent after showing that the meat could be preserved and it was placed in the glass bottles and boiled. This was the beginning of the food preservation. And in 1837, Swan demonstrated that heated infusion remained sterile in the presence of the air. Heated infusion can remain sterile in the presence of the air. And again, it was used to disapprove spontaneous ingestion. And it changed the effect of the air. In 1857, Louis Pasteur showed that the sour milk was caused by the microbes. And in 1860, he demonstrated that he destroyed undesired microbes in the wine and the beer. And later, the process is known as the pasteurization, which is very important till now. So the microorganisms which arise in the field of the microorganisms are bacteria, okay, bacteria, fungi, protozoa, algae, and the viruses. Bacteria, fungi, algae, protozoa, viruses. So these microorganisms will be discussed in the field of the microorganisms and in next lectures also. What are their cellular organization? What are the microorganisms? What is their use and what kind of cell organelles are present in them? What is their basic structure? What is their classification? What are the examples? So it will be discussed in next lectures. So according to this lecture, the question which can arise, explain history of the microbiology. So students learn a lot from this lecture that how what is the history, what different kinds of theories were given in the field of the microbiology at the starting phase. What was the preservation methods in the early stage of the microbiology? How the generation, the starting generation, use methods to preserve the foods, and what were their studies in the microbiology, and what were the studies uh, to ferment, what were their studies to production of food by the fermentation process. So these are the methods. So my topic is over here. Next video I will discuss another topic. Thank you very much.